Hi, everyone. My name is Kyle Morris, and I'm the Chief Scientific Officer here at Solid Bioscience. Thanks, everyone, for taking a look at this presentation. I hope it's helpful. I'm providing an update of our AAV microdystrophin gene therapy program, SGT-001, here at Solid Biosciences. As a public company, before I get going, I have to highlight the forward-looking statements. And importantly, I think I want to acknowledge and recognize the unprecedented time we are all trying to navigate through right now. It's important in this community to really stay strong and committed and work together. Solid's mission is unchanged as we remain 100% focused on our commitment to the Duchenne community, regardless of the global circumstances we all find ourselves in right now. We're working with the same drive and scientific rigor as ever to resolve the IGNITE DMD clinical hold, while keeping patient safety as our top priority. As I'll discuss today, we're making progress with our gene therapy program, SGT-001, and are working hard to push things forward. Hopefully we'll get through this together. To make this presentation as real as possible, I won't be stopping recording when I make my typical mistakes, might say my arms and use the wrong words. Hope that's okay with everyone. So just want to introduce the microdystrophin gene therapy program using a clinical candidate, which we describe as SGT-001. So with our AAV gene-mediated gene therapy, there's three specific requirements or components: the AAV vector, the transgene of the protein of interest that we're trying to get expressed, and the promoter that is used to drive the expression of that protein. In SGT-001, each of these components was really carefully selected. Starting from the bottom, AAV9 was really chosen given the prior clinical experience of this vector and also the known muscle tropism of the serotype. We thought it was a good choice to move forward with. The CKA promoter was developed by Jeff Chamberlain and Steve Hauschka using both in vitro and in vivo models to identify what's known as a regulatory cassette that can produce high levels of protein expression in muscle. And what we found is the CKA does that quite well and is very, very specific. We have not ever seen expression of the protein in any other tissues other than muscle tissue, both cardiac and skeletal muscle. The transgene for our microdystrophin, a smaller version of the full-length dystrophin, was rationally designed and developed by Jeff Chamberlain after many years of iterative research and experimentation to optimize for both expression and function. Additionally, an important aspect of our current construct was the inclusion of the nNOS binding domain that I'll describe a little bit, first identified and combined with with Jeff Chamberlain's work by Dongsheng Zhuan. I'll talk briefly about the overall microdystrophin construct on the next slide. What we're showing here really is the protein domain structure of the full-length protein dystrophin. It's a large protein, and the DNA sequence required to produce this protein is almost 3 times too big to fit into the AAV vector. So what needed to happen was really there had to be a rational design approach to really determine the portions of the dystrophin molecule that were needed to maintain a similar level of function as the full-length protein, but at approximately 1/3 the time, or 1/3 the size. Jeff Chamberlain and others, over years of work, were able to determine what pieces were necessary. There are 24 repeats in this protein, most of which appear to be redundant, but it took a lot of work to tease out this understanding. Ultimately, what we came up with at the bottom of the slide, the SGT-001 microdystrophin protein, was designed, tested, and shown to generate a highly functional protein that was consistent with wild-type levels in some mouse studies that I'll show. Dongsheng Zhuan found that the repeats R16 and R17, highlighted in the middle of the protein, are actually required to add the additional functionality and recruit in the nNOS binding. So they recognize as the nNOS binding domains, and I'll talk about those a little bit further as well. So once the components were selected and our construct was put together to make SGT-001, we tested the level of microdystrophin function in both dystrophic mice and in dystrophic dog models. Here, what we're showing on the slide is on the top row, a dose-dependent increase in microdystrophin expression in a muscle cross-section in NDX mice by immunofluorescence. Top row is heart, and the bottom row is skeletal muscle. The microdystrophin has been labeled with a specific antibody and shown with a red staining around the outside of those muscle fibers. What you can see is we get a really nice dose response going from the dystrophic level, where there's none, very little, to at the high levels, 5e13 and 2e14 vector genomes per kilogram. You see really nice, consistent, widespread expression in the mouse heart and the skeletal muscle. Shifting a little bit to why we think nNOS is important. Really, the role of dystrophin appears to be primarily to be as somewhat of a shock absorber, linking the contractile sarcomere structures, the actomyosin protein structures, together with the membrane, together with a protein complex that's put on the membrane. This protein complex is known as the dystrophin-associated protein complex, or the dystroglycan complex, and this includes proteins like beta-sarcoglycan and sarcospan, as shown in the figure. Additionally, it also functions to bring together other proteins and localize them near the surface of the membrane. One of these proteins is known as neuronal nitric oxide synthase, or nNOS. Without dystrophin, the nNOS does not localize to the muscle membrane, and the dystrophin-associated protein complex isn't formed. And that can be shown on the left-hand side, where in the absence of dystrophin or microdystrophin, you don't see NO production or the protein complex formed. While on the right-hand side, with the addition of either dystrophin or, in our case, microdystrophin, you see the additional benefits. What this means is, and so what's important about this is that nitric oxide synthase, or nNOS, produces nitric oxide, which in turn allows for vasodilation, so increasing blood flow, and therefore allowing more oxygen to get to the muscle when it's needed. So during activity or exercise like walking fast or running, with dystrophin localizing nNOS near the membrane, allows for the natural response to the contractions, reduces and induces the switch on the NO production, dilate the blood vessels, and get more oxygen into the muscle. In Duchenne, the lack of nNOS means there's a limited ability to increase the blood flow and get the oxygen to the muscle, generating what is called a functional ischemia, or a lack of oxygen in the muscles. By incorporating the nNOS domain into our microdystrophin construct, we have restored the ability of the protein to recruit nNOS and therefore produce the nitric oxide that will allow for increased blood flow back to the muscles, potentially reducing the effects of functional ischemia. So importantly, there's preclinical and clinical evidence. There's more than I'm showing, but I'll just sort of highlight two aspects of this that show the benefit of including the nNOS binding domain. In MDX mice following SGT-001 administration, force generation in levels in the diaphragm were found to be comparable to that of wild-type mice, and significantly greater than the force production in untreated mice. And that's shown on the left-hand side, the left-hand panel there. In Becker's muscular dystrophy, dystrophin protein is expressed, but it's generally at low levels and is a truncated version of the muscle, of the protein, and it's based on the mutation of the individual. When we evaluated a natural history cohort of Becker muscular dystrophy patients, and an association between the patients having a complete nNOS domain, highlighted here, having a complete nNOS domain versus those that did not have a complete nNOS domain, we found that we could actually sort of distinguish a difference in the nitric oxide levels. There's a reduction when you do not have the nNOS binding domain present. And this is also in the DMD cohort as well, showed lower levels of nitric oxide in the blood. Also, and somewhat interestingly, there's a improved function as well. So in the 10-meter walk-run time function test, patients with the nNOS domain had a faster, an increased velocity or a faster speed than those that didn't have the nNOS domain. So it's sort of small indication that there's a potential association between nNOS binding, NO production, and potentially a functional improvement that can be identified clinically and provide significantly more benefit for the patients. So there are more activities underway to better understand the potential benefit of the nNOS domain for Duchenne. We're starting to explore ways to evaluate physiological changes in the SGT-001 dosed subjects. Now I'll transition over to our clinical study, what we call Ignite DMD. So Ignite DMD was originally designed as a phase 1/2 dose escalation study in Duchenne patients, and we had two separate different groups. First was an ambulant DMD cohort, that would be aged 4 to 11, or non-ambulant DMD aged 12 to 17. And we made that distinguish somewhat arbitrarily to separate out the ambulant and the non-ambulant. We went in, and as shown here, we went in with on the left, at first with a dose of 5e13 vector genomes per kg in a non-ambulant adolescent. After that, all subjects that had been dosed in the study had been children. A total of 3 subjects were dosed at 5e13 before the planned dose escalation to 2e14 that occurred between subject 3 and subject 4. Now 3 subjects have been administered SGT-001 at the high dose level of 2e14 vector genomes per kg. The primary endpoint, as with all phase 1 studies, is safety and tolerability. However, we also included microdystrophin expression at 12 months as a primary efficacy endpoint. Secondary endpoints within the study included the standard functional assessments you're well aware of, such as the North Star ambulatory assessment, the performance of upper limb, and even the 6-minute walk test. We're also evaluating respiratory function and examining muscle and heart quality via MRI. The study includes other activity monitoring approaches, and generally we are using this study to try and collect useful data for the appropriate inclusion into possible future studies. As noted, the study is currently on clinical hold and will require FDA agreement, as well as both Drug Safety Monitoring Board, DSMB, approval and IRB approval at the study sites to reopen the study and begin dosing. So to summarize where we are with the IGNITE DMD study, a total of 9 subjects have been enrolled in the IGNITE DMD with 6 dosed with the SGT-001 candidate. Currently, all study activities have been completed at the University of Florida. I think most importantly, all 6 dosed patients are doing well and continuing the study per protocol. The aim, given the current circumstances, is to ensure patients that are currently enrolled in the clinical study continue to be evaluated as closely to study protocol as possible, while of course safeguarding their health and well-being in the midst of this pandemic. As mentioned, the study is on clinical hold. We're actively engaged with the FDA to resolve the issues and again move forward. Prior to the clinical hold, we had announced that we had opened 2 new sites, one at UMass Worcester and the other at Lurie's Children in Chicago. Will provide with more options for dosing when the hold is lifted. We were able to obtain 3-month biopsies from all 6 dose subjects, and based on these results, particularly from the 2e14, provides confidence in the program and belief that we should move forward and continue dosing after the appropriate approvals have been obtained. I'll talk about them now. So what did we see in the 3-month biopsies? So the results were positive and really show the potential of SGT-001 to provide meaningful therapeutic benefit to patients with Duchenne. Evaluations of muscle samples using immunofluorescence, like I showed for the MDX mice earlier, highlighted broadly distributed microdystrophin expression throughout the entire muscle sections in all 3 high-dose subjects. The percent of microdystrophin positive fibers ranged from 10 to 20% in subject 4 to 50 to 70% in subjects 5 and 6. Positive fibers were found to be widely distributed across the section, not localized to small regions of the muscle. And this is important as it shows that the AAV is getting where we want it to, and that is to all of the muscle. As I mentioned before, dystrophin functions to stabilize the dystrophin-associated protein complex and nNOS out on near the muscle membrane. To show functionality of our microdystrophin protein, we look for the co-localization of beta-sarcoglycan, one of those membrane complex proteins, and nNOS in the muscle biopsies from the patients, and found clear evidence of both proteins being stabilized in the microdystrophin-positive fibers. We assessed nNOS activity as well using an enzymatic stain. We're able to show positive staining in the microdystrophin-positive fibers. When we quantified by Western blot using our qualified method, we found that the 2e14 dose produced between 5 and 17 of normal levels of dystrophin. And we'll continue developing our method to better understand how that fits. We also have mass spec, and we have consistent results from that assay as well. The overall results from the biopsy analyses indicate that SGT-001 is distributing throughout the muscles and producing good levels of microdystrophin positive fibers that show functionality via the co-localization of both beta-sarcoglycan and nNOS proteins that we've measured. Given these initial results, we continue to push forward and really want to get back into the clinic as soon as possible. So given the promising preliminary data, I think it's important to look at how we're going to produce and manufacture SGT-001. Taking one step back, Duchenne really represents presents a significant challenge in manufacturing, given several factors: the high number of patients, so the prevalence, the need for high systemic doses, and the average weight of the patient that requires treatment. All of these combined mean that a significant supply is required to administer gene therapy, AAV gene therapy, to the Duchenne community. There's massive need to produce sufficient drug to dose the entire population, and it really requires a truly scalable process to meet those future demands. We selected an HSV-based approach, a scalable suspension method to help address the anticipated need. So where are we? We have, over the past few years, really been focused on building consistency in our process while scaling up to levels capable of supporting the needs of Duchenne. Currently, we've produced multiple 250-liter batches with our CMO partners, allows for the dosing of a number of patients. The process itself uses widely available and standardized technology and equipment, and therefore allows for mixing and matching and good tech transfers when needed. This suspension-based approach also provides the opportunity for further scale up from our current 250-liter, potentially up to a 2,000-liter scale if necessary. We, while maintaining the same process, fundamental process. We continue to further optimize the process to help address the needs of the community. So overall, in summary, I think you can imagine our clear and immediate goal is to work with the FDA, DSMB, and the clinical site, IRB, as quickly as possible and resume dosing safely and responsibly. We're looking at all aspects of the program to identify areas to optimize or improve our drug and our study to allow for a safe and effective administration of SGT-001. So to wrap up, we are excited and optimistic about the promise of SGT-001 to provide meaningful benefit in the lives of DMD patients across the disease spectrum, and of our ability to supply SGT-001 to meet those patient needs. With the biopsy results indicating widespread microdystrophin expression and nNOS activity in the muscles, we want to get back into the clinic safely and move forward with SGT-001. We continue to follow the current study subjects per protocol and are looking to evaluate samples and the functional endpoints from the 12-month time point. These, of course, being done with consideration of the current environment. We are hope to really be sharing updates regarding the study over the coming months. And so with that, I'd like to close by expressing our deep appreciation to all the patients and the families and at the University of Florida who have generously participated in the clinical trial so far. And to all of you for your attention. Hopefully this was of value. Stay safe and be well. Solid is going to be here for Duchenne, and we'll get this through together. Thank you very much, and goodbye.